Good morning, one and all. I'm Dr. Aarti Venkatesan from the team of LLB School. I welcome everyone for another CPMB LLB web series. And we welcome our speaker, Dr. Bony Kumar, Director from Genomics Core. Now, I request Professor Jnanam, Head of Bioinformatics Department, CPMB TNAU, to give a brief introduction about our speaker. Jnanam, ma'am.
Yeah. Good morning to everyone, and a special welcome to Dr. Bony. I just introduced Dr. Bony, a very brief introduction I would like to give. He has completed his BSc Agriculture from Kerala Agriculture University during 1995 to 2000, and later he did his MSc Biotechnology at TNAU, CPMB TNAU during 2001 and 2003, where I was fortunate to guide him as my first MSc student. Later, he moved for his PhD to Life and Biosciences at the Open University, UK, during 2010 to 13. After his PhD, he has joined for his postdoctoral fellow at Stowart's Institute for Medical Research in Kansas City, USA. Then he has joined as a genomics scientist in Jackson's laboratory, Bar Harbor, and currently from 2018 onwards, he is the director, Genomics Core, School of Health and Medical Sciences, University of North Dakota, Grand Forks. This is the only school of medicine which is there in the state of North Dakota. And in 2018, Bony was offered with two choices. One is with the Columbia University in New York, and the other UND School of Medicine and Health Science. He has chosen the latter, and he is happy with the choice of what he has made. This is an interdisciplinary research center which is originally financed by National Institute of Health as a center of biomedical research excellence, and this is dedicated to provide state-of-art genomic resources, which includes technical training, data analysis, grant assistance to investors, and this will also take care of the external commercial clients. His main vision is to train people, let that be principal investigators, postdoctoral fellows, graduate students, or even the medical students, so that ultimately they will come to know like how they can use this in their own work when they end up. That is his main motto, and to his credit, he had a very good number of publications, particularly in peer-reviewed, high-impact journals like Genomic Research, Nucleic Acid Research, and Natural Communications. With this brief introduction, I request Dr. Bony to give his special lecture. Thank you.
Yeah. Thank you, ma'am, for the introduction. Sir, can we start with the session?
Yeah. Thank you so much, Jnana madam. Actually, it's really, really great pleasure to be, hearing you also after a long, long gap. When I was actually given a choice for a talk, I thought that it may be good to just talk about where are we with sequencing technology. And the reason why I thought the topic may be relevant because every day sequencing is something that is actually changing its landscape in terms of technology, in terms of cost, in terms of the capabilities of this instrument. So I thought it will be a nice, really nice time for us to talk about where this whole story started, story of sequencing, and where it is standing now, and what we are looking into when we are looking into the future, where we are actually going to see ourselves in, say, another 5 years or so in terms of genome sequencing. So before that, let's think about a little bit more historical perspective of DNA, about the molecular biology in general. So if you think about biology in general, it was a descriptive science. So descriptive science means that you are basically seeing something, opening it up, describing it, and that's it. I mean, that is what actually biology was till discovery of DNA or discovery of molecular biology. So what really happened in, like say, 1970s and afterwards, that we started manipulating DNA. We have started cutting, joining DNA, and that led to a whole new field of study called as recombinant DNA technology, which in turn made biology from a descriptive science to an experimental science. So we started actually manipulating organisms, and then we started looking at effects of those manipulation, and that was the beginning of all the science of experiment. And so DNA played a big role in understanding of DNA played a big role in transformation of biology from experimental science to a descriptive science to an experimental science. So if you think about history of DNA, it actually not just something it's not very new. Actually, DNA was discovered way back in 1869, and it was chemically characterized in 1909. But 1953 was the breakthrough year when the first time structure of DNA was solved, and it was actually known to the world that it's a double helix structure, the famous Watson and Crick experiment. In 1977, the first Sanger sequencing was invented, and we will actually looking into this more in more detail in later slides. And then first genome was sequenced using Sanger sequencing, which was a fast DNA, which is 5x174, a very small fast DNA, 5 kb long. In 1986, first automated sequencing machine was made available in the market, and 1990, Human Genome Project started. In 1992, first sequencing factory was established, which was to tackle human genome sequencing project. And in 1995, first complete bacterial genome, which was influenza bacterial genome, which was around 1.8 MB big, was sequenced. First animal, which was sequenced, was C. elegans, which was around 97 MB big genome. And in 2003, we completed Human Genome Project, which is till this time, I think that's the biggest genome we sequenced, around 3 GB big genome. And it took almost 13 years and $2.7 billion US. So this number is significant. We will come back to this number in a couple of slides later, that why this number is so significant. So it took almost 13 years to complete human genome sequencing, and at an expenditure of around $2.7 billion US. And then in 2005, first next generation sequencing instruments came into market. And by 2013, we have more than 10,000 genome sequences available in NCBI databases. So, but what is actually main thing happened during all these years from 2005 to 2025 or 2020? So you remember I told you that Human Genome Project, it took 13 years and $2.7 billion to sequence. And if you look at the cost of sequencing now per genome, is actually down to almost $800. So in my facility, we sequence whole genome in a night time, so morning we will start. If we start sequencing run by next day morning, we have a whole human genome sequenced at least 3 times, that is at least 3x coverage, just for under $800. So think about 13 years versus a night, and 2.7 billion versus $800. So where are we expecting it by 2025? So the idea here is that the sequencing cost per genome should drop down to $1, just for $1, you should be able to sequence your genome. And why that is important, if we are able to achieve even not $1 landmark, but even if we could achieve around $100 or $200 genome, $200 per genome sequencing, it will open up floodgate of personalized medicine. So if you haven't heard this word called as personalized medicine, then personalized medicine means that doctors will make a decision about your therapeutics, what kind of drugs you need to be given, or what kind of tumor is growing in your body, all this through sequencing. So they will do a genome sequencing, and based on the sequence information, they will choose what drugs will work in you, what combination of drugs will work in you, which kind of drug will not work in you, all those kind of decisions. So to really make that happen, the sequencing cost should come drastically down. And even at $800, it's not a bad deal nowadays. But if it comes to $100 or $200, everybody will be using genome sequencing as a tool to make therapeutic decisions. So the idea or the expectation is that by 2025, we will be able to achieve $1 genome sequencing. But the way that technology is growing, I don't see that's going to happen by 2025. Probably we will be touching a mark of like say 200 or $250 per genome. So this kind of growth was actually all started with first generation of sequencing. So what is the first generation of sequencing? So first generation of sequencing was actually based on a couple of different technologies that was developed independently by many different people. So one was chain termination method, which was discovered by Sangers and Carlson in 1975. The other method was based on chain degradation, which was developed by Maxim and Gilbert during 1976-77 period. The third technology was pyrophosphate sequencing, and fourth was ligation enzyme method. So the pyrophosphate sequencing was a first generation sequencing invention, but there were no this method was not that popular during first generation sequencing. Later on, in the next generation sequencing platforms, they started using pyrophosphate basic methods, and so now there are methods actually available. So you can actually, there are machines that are available that uses pyrophosphate based sequencing methods. So what is actually the advantage of this first generation sequencing methods? So the read length ability was only 100,000 base pair, but it can actually do that with very high accuracy. So the base calling or the base accuracy was more than 99.99%. So that makes this method very, very interesting as well as usable. And whole genome, first human genome was sequenced using first generation sequencing approach. And now also, we actually do a lot of this first generation sequencing for validating the PCR products or validating various cloning approaches that we various clonings that we do within the plasmids and all. So what is actually a real disadvantage of this first generation sequencing method? The first generation sequencing, the biggest disadvantage is its high cost. So if you want to sequence whole genome using first generation sequencing, as you saw in case of human genome, it will cost a tremendous amount of money. So like around $2 billion, $3 billion to sequence. Even today, if you want to sequence human genome with first generation sequencing platforms, it will cost almost that much money. And other thing is low throughput. So throughput is very low. That is why it took 13 years to sequence one human genome. What happened? Oh.
Hello, sir. Is there any issue?
Yep, I am not able to move my slide for something. You can, it's saying that you can annotate and then.
Okay. You have turned on the annotation actually. So there will be option below.
Stop annotating. Okay, I think that's okay. Great. Okay. So yeah. So in case of Sanger sequencing, what we actually do, we add different nucleotides, and so basically, there are 4 types of nucleotides in the DNA. So DATP, DCTP, DGTP, and DTTP. So basically, deoxy variants of ATP, CTP, GTP, and TTP. So if you actually remove 2 oxygen group from this ATP, CTP, DTP, GTP, and TTP, it will become DDTTCP or DDCT. So whenever that particular dideoxy nucleotides are incorporated, chains are terminated at that position. So as you can see in case of this image, can you see the cursor here? Okay. So what happens when the base actually got incorporated and it terminates the chain? For at each position, there will be one termination event, and that termination event is you can see that which base is actually responsible for that termination using fluorophores that is actually present at that position. So each DDoxy nucleotides are actually labeled with one particular fluorophore. And then once you do the electrophoresis on a capillary gel, and the detector can detect that those bases, and then based on that, you can recreate your sequence. So this was actually a whole basis of Sanger sequencing. So I was telling that there are a couple of 4 different methods where they're developed during first generation of sequencing, but Sanger sequencing was the only one which could get automated. So automation was possible only because of Sanger sequencing. That is why the first sequencer, automated sequencer, was developed based on Sanger sequencing. And these Sanger sequencing based instruments are still working, and they are widely used in all the different setups. So that so as you saw, that the biggest challenge for the first generation of sequencing was its cost. So you need actually you have to spend a lot of money to get things sequenced, and then as well as there was a huge throughput problem. So it's a very low throughput method. So for that, the next generation sequencing technologies started emerging in 2005. And that was actually based on, there were 2 different generations of next generation sequencing after 2005. One was second generation sequencing, which was based on short read sequencing platforms. And so there were 2 different methods, like sequencing by synthesis or sequencing by ligation. And the second, third generation sequencing, which was basically long read sequencing platform, and where the sequencing was based on either synthetic long reads or single molecule sequencing. So we will see each of this platform in more details as we actually go. So let's think a little bit more about second generation sequencing now. So there are certain terminologies that you should be familiar when we are discussing this about the second generation of sequences. So one is called as type of reads, or a read is a sequence, inferred sequence of base pairs corresponding to a single DNA fragment. So whatever you see, a single base pair of DNA, whatever you sequence, that is called as a read. And the second word is library. So you have to prepare your DNA fragment suitable for sequencing. And that pool of material or DNA, which is modified in such a manner to get it sequenced, is called as library. And the third is coverage or depth of sequencing. So how many times a sequence can cover whole genome, that is called as coverage. So suppose you sequence so deeper that you can, all the sequence can tiled and it can actually cover 3 times whole human genome, then the coverage is 3x. So when you are thinking about genomic experiments or design, you have to think about what are the kind of reads that you are looking for, what kind of library you are going to make, what different coverage or depth of sequencing you need, and what platform you are going to use. So there are different kinds of reads. One is called a single end sequencing, that you sequence from one end, and that can be randomly chosen between 5' end of a DNA or a 3' end of a DNA. Or you can do paired end sequencing. Basically, you are sequencing from both ends. You are sequencing from 5' end, and then you sequence from 3' end. And there is another kind of libraries which are actually used in a very specific case, which is called as mate pair sequencing, where the ends are actually, originally they are far away regions and they are sequenced together. So there are uses of that. So don't worry about that at this point. But what we have to think about that for the time being, you should know that there are you can either sequence a library from one end, or you can sequence library from both ends. So a general workflow of any NGS or next generation sequencing is that you take your DNA. So if it is a whole genome or a fraction of DNA, whatever you are taking, you fragment it into small pieces. So your piece is actually somewhere between 100 base pair to 600 base pair. And then you amplify each piece into thousands of or millions of molecules. And then each of this molecule, you parallelly sequence them. So you sequence them in one single lane or one single channel, and you will generate a huge, massive amount of data. And that data will come out of the instrument. And then there is a process called as base calling. So each of these instruments, you will see that they generate a different kind of signal. And that signal needs to be converted into nucleotide bases, or like ATGC, because that is what we understand. So that process is called as base calling. And once you do the base calling, and there's a whole battery of analysis goes through at the bioinformatics end to create file that is actually usable for the analysis. So this is the general workflow for sequencing. So you take your DNA, you share it, then you amplify it, then you add few sequence at the end of it to make it suitable for sequencing. Then you sequence them, and once the sequence, whatever in output of your sequencer is, convert that output into base pairs through a process called as base calling, and then you convert your file into a more amenable format, which can be used later for different kinds of bioinformatics analysis. So, so let's actually think about what are the different NGS platforms that are available. So there are different. So these platforms differs in designs and chemistries, and sequencing of millions of actually clonally amplified molecules are done in a massively parallel manner on these platforms, and they have a lot of actually clinical application. But the analysis of some of these are actually a little more complicated than just sequencing one single fragment of DNA. So these are some of those platforms that is actually available. So one, I was telling that there was a first generation sequencing technology called as spiral sequencing, which was not used much in first generation, which was later used in second generation sequencing. And Roche, a company, it actually developed a platform called as Roche 454, and that uses this spiral sequencing based technology. And there's another company called as LifeTech. It developed a platform called as Solid, and this is basically sequencing by ligation. It uses a method called as sequencing by ligation. And then you have another method, other instrument developed by LifeTech itself called as Ion Torrent, and it uses H plus hydrogen ion based estimation. And then the leader of sequencing technology, or market leader of sequencing technology, is Illumina. And it uses a method called as reversible dye terminators. Almost 80% of sequencing market is currently captured by Illumina. Illumina is actually a market leader in this. How most of them differ. So they differ in terms of what kind of signals are the output, and how they actually amplify clonally. So for all other methods except Illumina, the clonal amplification is done through a method called as emulsion PCR. So basically, you incorporate your DNA into one single oil emulsion droplet, and along with the reagent for PCR amplification, and amplify that particular fragment of DNA inside that oil emulsion. And this method is called as emulsion PCR. So all other methods except Illumina uses ePCR for generating clonal amplification. Illumina uses another method called as cluster generation, and we will see what actually that method is slightly shortly after this. So output from these instruments are different. So pyrosequencing, the luminescence is what actually read as our output. In case of solid, fluorescence is used as a method to detect the signal. And in case of ion torrent, it's a pH change, what is actually used as a method for detection of bases. And in case of Illumina-based reversible dye terminators, it's the fluorescence that actually uses a reaction output. Basically, whatever signal to noise ratio is there, that is what actually converted into sequence, and that process I explained to you called as base calling. So in case of sequencing by ligation, what happens that you have a two base pair fragment, very defined two base pair is actually added at the end of a primer, and that is actually ligated, and then reaction is extended, and the base. So that actually, whichever base is basically added, and that makes a complementary pairing at that is considered as the base at that position. So that by ligating and then releasing the ligation and again religating, this whole sequencing process is actually goes from one end of the DNA to the another end of the DNA. So here so basically instrument adds dinucleotide one set of dinucleotide at a time, and then wash it away, then add another set of dinucleotide, and then extend it. So that is a general process how this is. This whole process is actually done through ligation. In case of ion torrent, what happens is that one nucleotide is actually added at a time. So like in this case, you have AGC, and when T nucleotide, which is actually based complementary base to A, comes into this channel, then what happens? It makes a complementary base pairing, and that releases an hydrogen ion. So the release of hydrogen ion creates a slight change in pH, and there is a pH sensor at the bottom of this channel that senses that pH change. So once that pH change is there, now machine knows that, okay, you added T at that time that pH change happened. So the base sequence at this position is A. So like this, actually, based on pH change, ion torrent senses what is the reverse complementary base, and then that actually is recorded as a base in case of ion torrent system. There is another interesting technology for identification, for quantification of gene expression, especially, is called as nanostring. So what you do, this is a mid-output way of sequencing. So what basically you are doing here, that suppose you have, say, you want to know the expression level of a particular gene. And so you make a reporter probe, which is complementary to your gene, and then hybridize your probe. And then you wash off unhybridized probes, and then count how many molecules with fluorescence is there in your system. So you are basically counting how many probes are reverse complemented to your target region, and that count will give you a number of molecules or number of transcript for a particular gene. So this is basically used for gene expression analysis, and it's actually pretty popular in case of clinical setup where you have a panel. Say in case of cancer, you have 50 genes or 60 gene panel, and you want to just focus on those expression of those 60 genes. So where the nanostring is pretty useful. And that good part of the nanostring is that you don't need too much complicated bioinformatics analysis for resolving your data set out of the instrument. So, but as I said, like the market leader for sequencing technology is basically Illumina, and it actually comes with all kind of flavors. There are instruments called as mini-seq to MySeq, NextSeq, HiSeq. You can see all the seqs are there. So the latest one of this series is NovoSeq, and it can actually give you, from one flow cell, it can give you 2.6 billion reads. That means basically what you are and then currently in US, one lane cost around $7,000. So basically a huge amount of data for a very small amount of price. So which basically makes this whole system pretty attractive. How actually Illumina works, as I was telling, that it does something called as bridge amplification. And so when I was telling you that you need to prepare your DNA for sequencing. So you add some extra sequence at the end of the DNA, and those sequences are called as adapters. So there are, and there is a glass slide is there, which is used in case of Illumina sequencing, and that glass slide is called as flow cell. So on the flow cell, there is a mat of small short oligos, and those oligos are complementary to the adapter sequence on your DNA fragment. And so they actually base pair on the glass floor like that. And then you actually, so that will, and then the other side of your DNA molecule will go and reverse and then again cross, look for the complementary sequence, and then it will make an arch-like structure. So this is what called as bridge. And then what happens? A synthesis reaction happens, and second strand of this molecule is synthesized by polymerases, and in this process actually, and then they make whole bundle of sequence out of this. So basically this is how they do the clonal amplification. And And this multiple cycle of this goes ahead, and then it makes that whole bundle of amplifiers, which is important for improving signal to noise ratio. And once that is done, then what happens that one base is added at a time, and each of these bases are labeled with different fluorophores. And once the base is added, and it actually makes a reverse complementary pairing here, then after the washing, the instrument takes a picture of that. So basically, if you have 200 base pair long fragment, it will take 200 pictures, and it will stack that picture over one over another, and that pictures are actually converted into those pictures are actually converted into a sequence. So that is how it looks like. So this is how it looks like. See the picture from flow cell. So this is a glass flow cell, and it has small chambers in case of pattern flow cells. And this is how. So this base is green, so maybe T, I believe. So green is C. So the green one is C, and then if you have a then it should be red. So basically, by seeing these dots, you can figure out which sequence is which, and that is what the base callers do in case of Illumina. So this is one of the most popular technology in case of sequencing, and as I said, almost 80% of flow sequencing is basically is actually controlled by Illumina. So that brings me to the third generation of sequencing platforms. So before that, as you may be like thinking, like, as I sounds like that, I'm so happy about second generation platform. They give so much data. It's very cost effective. Then why we need long reads? So why we need a next generation of sequencing platforms? So before to think about that, why we need long read sequencing, think about a puzzle. If I give you a puzzle which is say 10,000 pieces or 92,000 pieces, okay, so if I give you that puzzle to join and make a whole picture out of it, how difficult it will be. But same thing, if same puzzle is actually just 9 pieces long, you can basically combine them and make a beautiful picture, as actually shown an example here. So that is the advantage of having long reads. So basically, if you have a long read, you can assemble them easily. And so apart from that, there are, like, especially in there, those who are in agriculture, you know that a lot of these crops are polyploid. So assembling polyploid genome with short reads are very, very difficult. So assembly of complex genome is actually easier with long reads, as well as those areas of genome which has high amount of repetitive elements, short reads cannot assemble them. So long read sequencing platform can actually help you to assemble short read, repeat regions also from the genome. So here is one example, like you generate only 16 GB of data and you can actually make whole, so at the 6x coverage, you can actually create whole wheat genome, which is around 16 GB. So a lot of these polyploids like potatoes, canola, cotton, sugarcane, wheat, all of them can be actually very successfully sequenced and assembled using long read sequencing platforms. So how you generate long read? So one way, you can use short read as a way to create long read also. So this is called a synthetic long reads. And how you create synthetic long reads? So basically what you do, you put your DNA, long DNA, into one small microfluidic chamber. You fragment them inside those microfluidic chamber, and then add a barcode or a tag at the end of it saying that all these molecules are coming from same microfluidic chamber. So basically what you are doing, that each microfluidic chamber has a small tag associated with it. So and once you fragment your long read into fragments, you add those small barcodes or indexes into that, and then you pull everything, sequence them through short read. And when you are doing bioinformatics, you combine all the fragments which have same kind of a tag, which will give you a longer piece of DNA. So you are not you are still sequencing as short reads, but through the bioinformatics way, you are actually generating a long reads. And this kind of approach is called as synthetic long reads. One of the technology that actually uses this is actually provided by 10x system or 10x. What happens that you combine your DNA, enzyme, and oil droplet, and inside the oil droplet, this is your DNA. You fragment them into small pieces, add same kind of a tag, and then you sequence, and then you will get something like this, like a linked reads out of this. And this actually, this way you can actually create a synthetic long reads out of the system. But there are methods that you can actually create single long read using single molecule sequencing. And one of those two methods are actually through nanopore sequencing and PacBio system. So nanopore sequencing, as you can see, this is on my hand, and this is only this big instrument. It's a very, very small instrument. You can connect it through your laptop. And you can sequence. So you can sequence it anywhere. Those who are working in environmental biology and you want to do some sequencing near a pond or in a forest, anywhere you can actually use this instrument for sequencing. But this other instrument is a PacBio system, which is kind of a little bigger system. But both of them do something called a single molecule sequencing. So let's see what these systems do. So in case of PacBio system, there is a microfluidic well. In this microfluidic well, DNA is actually goes inside that, and then let's see this way. And then polymerase actually keep traveling on this DNA template. And so what happens each time it adds a base has a fluorophore on that, and this one and that fluorophore is actually recorded, and that will actually leads to that will actually convert it back into sequences. Okay, so 4 different colors are given to 4 different nucleotides, and each base once it is added, it actually machine records those colors, and that convert them back into those colors are actually converted back into sequence. And this is actually kept, and the instrument keep doing this for hours. Basically, it will start sequencing from here and then go all the way till here and then come back here and again keep going. It will do like that, and it will do so many times. And then you actually bioinformatically you chop these sequences, combine them, and create a consensus sequence, and that gives you a very high quality read out of these instruments. Okay, so this is basically sequencing using polymerase. And why we need to sequence this molecule multiple times? So you all are molecular biology student, and then you know that polymerase does mistakes when they actually polymerase usually does small mistakes. So those mistakes are there in the sequences also. So that is why when you sequence same molecule again and again and again and again multiple times, and then you take a consensus sequence out of that sequence, then those consensus sequence will be of very high accuracy. So every time when a polymerase is making a mistake, it won't make that mistake on the same location. So mistakes will be random. And based on this principle, consensus sequences are generated and very high quality reads are created using PacBio system. So there's another platform called as nanopore sequencing, which is another very interesting platform I was showing you on my palm. So what happens, there are biological membranes or biological pores through which DNA passes. So once DNA, and there is a voltage across this membrane, and each base, when it passes through that pore, there will be a change in the voltage, and that change is basically recorded. And those changes are very, very unique to each base pair. And that basically, and those voltage changes are basically converted back into base pairs, and that is how a nanopore does its sequencing. So it's basic, and again, as I said, like each base, but it's not like single base is basically read. It's actually it's 5 bases together are read in this case. So as you can see, the data actually shows you changes in voltage and fluctuation in voltage, and this is finally converted as a sequence, string of sequences. So so we have all these instruments have neural networks which actually runs on this data, and that actually give you sequence. So this is just the same representation. But before actually going to the next part of the talk, the nanopore is a wonderful system for many, many things. So it's not just about recording the sequence. This is the only instrument which can actually say that if there is a modification on the base or not. So suppose you are so cytosine is methylated, or cytosine is hydroxymethylated, or or adenine is methylated as 6-methyl adenosine, or as 1-methyl adenosine. These nanopore sequences can actually see the differences in terms of voltage fluctuation, and you can actually directly say that which base is modified and which is not. And second thing, nanopore is the only platform which can sequence RNA. So it can actually sequence RNA directly. So that is actually why it is so interesting, that because of this ability of nanopore sequencing, direct RNA sequencing, there's a whole new field of research called as epitranscriptomics is opening up, which basically looks at base modification in RNA. And why that is important? Many diseases like Alzheimer's, cancer, Parkinson, it is actually seen that RNA has modified bases. And why that is important from mechanistic point of view, because those modified bases impact stability of RNA, its transport, nuclear or cytoplasmic transport, or even translations are actually important. By rate of translation is impacted by modified bases in RNA. So whole new field of epitranscriptomics has been opened up because of the nanopore sequencing technique. So that actually comes to the next generation of sequencing. We are at the 4th generation sequencing platforms. But at the 4th generation sequencing platform, instruments are same, but the approach is actually changing. So there is no new methods, or there's no new instruments are coming. But what really happening is that we are started looking data at single cell level. So like suppose when it was our belief, say suppose in case of cancer, that is actually one of the best example I can think at this point. If you think from a cancer, from a general perspective, people believe that there's only one kind of cell in cancer. So cell just divide and then just it become cancerous. But that is not the case. Actually, if you think about this, a huge amount of heterogeneity is there in the cancer. And that is why when you treat a particular cancer with certain drugs, it works, or sometimes it don't work in one patient versus another patient. And other thing is that, like, you might have heard that this guy got cured, and then after 5 years there was a relapse and this cancer came back. And that is because some of those cells, which are rare in rare populations, lying within deep inside the tissue, that actually get unnoticed, and they actually come back after a few years. So that means that most of the systems, including cancer or immunogenic systems, or all of the systems, actually have fair amount of heterogeneity. So that how you will tackle that. So if you are using any other method, say bulk sequencing methods, it will actually look for aggregate of expression. Say RNA sequencing of a tumor you are doing, then it will actually look at what is the average expression of all the cells. So there is no ability for us to look at single cell level, that each cell, what is actually the transcription or are the expression profiles of each cell. So 4th generation sequencing basically is dealing with those issues, where you are creating data for single cell. So you have a whole range of tissue. You actually dismantle tissue into single cell, and then each cell you are sequencing separately. So that means that each cell will give rise to, and then you are collecting data from individual cells, and that data is basically analyzed to see that what is the kind of heterogeneity and all this kind of actually information you can. So this is actually the 4th generation sequencing platform, which is increasing the resolution of your sequencing from bulk sequencing to the single cell level. And that is what actually so basically one of the leaders of this system is called a Strenex Chromium system. And I'm just going to give you a brief overview of that. So what happens that there is a gel bead which you barcode, and each gel bead has a unique barcode along with. So each gel bead will have a unique barcode. Okay. Now it's a microfluidic system where from one end you will be passing these gel beads. From the second end, you will pass cells and enzyme. and the third there will be some oil. So what will happen that it will so you will eventually incorporate in a single oil droplet one cell enzyme and a barcode. And then inside this, all the reactions will take place. So whatever RNA or DNA is there in within this one oil droplet will have same barcode. And then you dismantle this oil droplet, you process your DNA and sequence it. Then what will happen that if you are looking at a gene, say here is an example where you have gene 1, gene 2, gene 2000, each of them will have same barcode, then they are classified as coming from one single cell. Similarly, if this other one will be classified as other cells. So like that, you can actually profile thousands of cells. And these instruments can profile on a single microfluidic chamber around 10,000 cells. So you can actually go up to 10,000 cells on this individual microfluidic chambers. And each of this instrument in one go can do up to 80,000 cells, profiling of 80,000 cells. So you do that, and then bioinformatically, you separate your data into cell-based analysis, and then go ahead and do this. There are other analysis called as clustering, and you do all those things and get your data at single cell resolution. So this is how it will look like. One of the example I'm just showing you that you can see like this is from a lung after the infection. They took the lung lavage and they wanted to see what kind of immune cells are there. And so you can see under two different conditions there are. So like in case of here, you can see the monocytes. So there is a whole expansion of monocyte in this particular deletion mouse line. So this way you can actually see what are the different repertoire of cells are there in a complex mixture. It can be any kind of cells. It can be blood, it can be tissue, it can be tumor, it can be lung lavage, anything it can be. And then you can actually look for how the cellular dynamics is changing and what actually that means from the biological standpoint. So just to summarize what actually, when you are doing a genomics-based approach or an NGS-based approach, think about what kind of reads you're looking at, what library you want to make, what kind of platforms you want to choose, and how many number of replicates or depth of sequencing you want to do. And that is very important because not for students, but for the PIs who are spending money. So they have to really figure out that how much money they have. And so the number of replicates and depth of sequencing has to do with amount of money that you have to spend. And then finally, what kind of downstream analysis you need to do, because a lot of bioinformatics goes into each of this technique, what you have seen. And a good bioinformatician can actually do wonders out of these data sets. Before I actually stop, I just want to actually show you where I am from. So this is actually, I'm from North Dakota, which is basically in the border of Canada and USA. Canada border is just 2 hours from where I am now. And this is my school, School of Medicine and Health Sciences, where I am. And my office is somewhere at the first floor at the bottom. This is how actually the inside of my place is. And so with that, I actually really like to thank a few people actually. So I'm really, I like to thank Gana Madam and family. Sir, both of them actually, when till the time we were there, they always considered as their family, and she was so good and she was so like graceful and helping. And sir also was like, so I always, used to think about them. Unfortunately, I never got a chance to again come back to Coimbatore, but next time when I will come to India, I will definitely come and visit all of you. And I'm really sincere thanks to Maheshwaran sir. I learned a lot of science from him and a lot of philosophy of science. He was a very different kind of person. And I still remember many of his quotes, especially one where he said that if you have a nail, a hammer, don't consider everything as a nail. He told that in terms of for marker technology. And I still remember that. And he was an amazing guy. And I like Sudha. I really, thanks to Sudhakar sir. He was the one who first taught us genetic engineering, and I still uses most of the cloning techniques, and I still remember him. Manikam sir, he was our head of department at that time, a great person. Kapil Dev Singh sir, amazing person. And then Thai Manan sir was there. I don't know whether he's still around. But his classes were still my inspiration. He taught us enzymology, and I still remember every single word of what he taught us. And I tried to actually imitate his style of teaching, but he was just like a magician on the blackboard. So I always miss his classes. And all of the teachers, Krishnaveni Madam, and there were so many others. I actually, all of them who actually greatly contributed to my career. So thank you so much for and your time and patience. And with that, I'm happy to take any questions.
Thank you, Dr. Bony. We have some questions. Shall I start with the first question?
Yeah.
Yeah. Could you please suggest tips to improve accuracy of sequencing the repetitive elements?
So it's not the problem of accuracy that is actually real issue with the repetitive element. It's the short read sequences gives you a fairly good amount of accuracy. But the problem is at the bioinformatic end, how you will align them back to the genome. So one of the approach like nanopore sequencing or even for that matter, PacBio does that. It actually you sequence larger number of times, like you sequence same sequence higher number of times. So basically, like go for a higher coverage. So if you go for a higher coverage, that will actually solve the problem with the accuracy.
Yeah. And question number 2. What is the accuracy rate of NGS techniques, especially in Illumina sequencing?
So Illumina is actually fairly high, 99.99% of a sequence. Now actually, so it started with 85% or so, but Illumina is almost at 99.99% in case of Illumina. But in case of Nanopore and PacBio, current accuracy rate is around 88% for the base calling. But when you use the method I called you, like consensus sequencing and as well as the coverage, if you increase your coverage, you can achieve up to 97% accuracy. And in Nanopore, there is another chemistry called as the whole new third generation sequencing, especially PacBio and Nanopore, you can achieve up to 96 to 97 to 98% accuracy.
And question number 3, what kind of approach we usually use while assembling polyploid genomes like wheat when we are looking or do a Jiva study?
So I think currently the cheapest and best way of wheat genome assembly will be Nanopore, in my opinion. You can still use short read sequencing, but I will basically suggest go for a long read fat form, and Nanopore will be best because it's instrument cost is less, and you can actually it does pretty good job. So I will go for Nanopore sequencing if I have to do wheat sequencing.
And the last question is, it would be great if we get to know how NGS can be used in microbial analysis and metagenomics from a bioinformatics aspect. That is whole workflow.
Oh, actually, I should have added something for that. Let me just. Can I have a minute or something? I can actually.
Yeah. Sure, sir. You can take your own time.
Yeah, I will actually show you something quickly. So I was not sure that anybody actually was looking for a microbiome based one. Okay, at least so here it is. Okay, I'm just going to show you this. So basically, for the microbiome, you can actually use either nanopore sequencing or short read sequencing platforms. So there are the basic idea here is that you have to sequence something called a 16S DNA sequence. Either so using nanopore or PacBio, you can sequence whole 16S region. In case of short read platforms, you will be using a portion of the variable region. So either like they have 8 different variable region, V1 to V8. So you will be sequencing either V2 to V3 and V3 versus V4 and some of those regions. So either you can use both of the technologies, but end of the day, you are sequencing 16S ribosomal DNA using one of those. So basic idea here is it is simple. You isolate your DNA from the sample. You PCR amplify your 16S region. So if it is nanopore or long read sequencing, you go for whole length of 16S DNA amplification. If it is Illumina based method, you actually amplify 300 or 400 base pair, one single variable region, and then you pull your amplicon, you barcode. So you give one barcode for your each sample, and then prepare library and sequence it. So basically, there are a set of primers that you need to use for differentiating different organisms, and then end of the day you will get something like this. So what are the different bacteria is there in, or so basically when you're doing this is for bacteria 16S. For fungus, there is another set of primer you have to use called as ITS primers, which actually amplifies variable regions from the fungus. And viruses, I don't think there is a kind of any platform that is actually currently available. I'm not sure about that. So that is how actually we do microbiome sequencing.
Thank you, Dr. Bony, for answering all the questions. I would like to request Jnanam ma'am to for the vote of thanks.
Hello, Bony.
Hi, Anna.
Yeah, I'm really excited to hear your voice as well as to see you after a long time. And it was, as usual, any discussion, he used to discuss in length about this. In the same way, I think whatever the lecture he has covered on NGS, it is regarding first generation, second generation, third generation, and including the fourth generation. He has given a very detailed account of all these technologies and where to use, how to use, and depending upon the budget and depending upon the need, whether it is going to be a single one, and the depth of sequencing, all the points he has covered very elaboratively, which shows his interest to teach the same thing to the participants of this particular webinar. I thank you so much, Bony, sitting late night and then explaining all the things to the particularly the alumni of CPME. And then thank you. And I thank LLB schools for making efforts in organizing this webinar and communicating to all the participants and to the individual students. Finally, I thank all the participants for sharing their valuable time in taking time to understand things, whatever was delivered by Dr. Thank you. Thank you.
Thank you so much.